Before we start, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present, and the elders from other communities who may be here today. Okay, so it's a pleasure to introduce Diana Hansen from the Division of Infection and Immunity. So Diana began her scientific career in Argentina, where she's from, and did a PhD at the University of Buenos Aires on Chagas disease in collaboration with the National Veterinary Institute in Uppsala, Sweden, and then moved to Australia. Despite the fact that she's been here for a long time, she still supports the Pumas when they play the Wallabies, which I still can't believe, being an avid rugby union supporter. But despite that blemish, she was, in her background, she was accepted to WEHI and did a postdoctoral fellowship with Louis Schofield. In 2010, she was appointed as a laboratory head in 2010, and her main contributions to the malaria field have been to understand the role of NKT cells in protection from cerebral malaria, and she's also shown that the chemokine receptor CXCR3 is responsible for recruitment of inflammatory cells to the brain in cerebral malaria, and has done work on IP10 and its role in that process. So Diana's talk is immune responses to malaria. Diana? Thank you.
Thanks, Alan. Can you hear me? Yes? So as Alan said, I'm going to be talking about immune responses to malaria. That is the work that we've been doing in the lab over the past 4 years. And in particular, looking at the role of inflammation, not only in the development of severe malaria syndromes, but also in modulating the acquisition of protective immunity. And I'd like to start the seminar with this map that shows the distribution of malaria around the world in 1942. So actually, I stole this slide from one of my PhD students. And this map is actually part of a pamphlet that was designed by Dr. Seuss when he was serving as a captain for the US Army during the Second World War. And the pamphlet was used to illustrate to the soldiers the risks of malaria and features Anne, or the Anopheles mosquitoes, and she drinks blood. So this is 1942. So this second slide here shows the distribution of malaria in 2013. So you can see the similarities are amazing, so, which means that despite a lot of research and effort, malaria still remains one of the most important global problems, with 250 million clinical cases each year and over 750,000 deaths. So from all the different species that infect humans, the most virulent is Plasmodium falciparum, which has many different stages in the mosquito and the human host. But for the purpose of this talk, I'm going to be mainly focusing on the intra-erythrocytic cycle of the parasite, because this is a cycle that induces the disease symptoms. And these disease symptoms could be very diverse, from starting from a mild fever or more severe complications, including respiratory distress, metabolic acidosis, renal failure, anemia, and in some proportion of the cases, cerebral malaria. Now, as a parasite immunologist, I believe that there are three fundamental questions yet to be addressed in the malaria immunology field. The first one is, what is the mechanism by which the parasite induces severe disease? Second one, why is that it takes such a long time to become immune to malaria? And last but not least, if it's possible to formulate an effective anti-malarial vaccine. So today I'm going to illustrate some work that we are doing in the lab, trying to address these three different questions, and I'm going to start with the first one, that is the mechanisms of pathogenesis. As I said before, cerebral malaria is the most severe complication of Plasmodium falciparum in humans, and the most susceptible population are children under the age of 5. And it's been estimated around 20% of the pediatric admissions still succumb to P. falciparum-mediated cerebral malaria despite administration of anti-malarial drugs, which illustrates the need for adjunctive therapies to treat this condition. Clinical manifestations include convulsions, impaired consciousness, and deep coma. And it is accepted that the disease results from the sequestration of parasitized red blood cells in brain blood vessels. Now, obviously, the analysis of these intravascular infiltrates in humans is only limited to having access to post-mortem samples. But we are fortunate that we have a reasonably good model to investigate this condition, that is the infection of mice with the parasite Plasmodium berghii ANCA. So there are different mouse strains that have different levels of susceptibility to this parasite. For example, C57 Black 6 mice are fully susceptible. They start to develop disease syndromes around day 6 post-infection, and they usually succumb before day 10 post-infection with relatively low parasitemia levels. And on the other hand, we have BAL-C mice that are resistant. So these mice do become infected. They actually develop high parasitemia levels, but in 90% of the cases, they don't get the cerebral disease. So this Plasmodium berghii model has been extensively used in my lab and a lot of other labs around the world to investigate the immunological basis of malarial pathogenesis. And basically what we have learned over the past 2 decades is that the early phase of acute malaria infection is characterized by a very strong pro-inflammatory response, which involves the activation of all these cells, NK cells, NKT cells, CD4 and CD8 T cells. So all these cells produce very large amounts of inflammatory cytokines like interferon gamma, and this is not such a bad thing because this interferon gamma is actually quite good at activating phagocytic cells like monocytes and macrophages, which are important to phagocytose infected red blood cells and control parasitemia. However, if this response is too high and becomes exacerbated, it could be pathogenic. Why is that? Well, happens is these inflammatory cytokines like gamma or TNF produced by activated macrophages are important activators of the vascular endothelium. And this basically results in the upregulation of adhesion molecules, which are involved in the recognition of parasitized red blood cells. So in other words, this cytokine storm exacerbates parasite sequestration in the brain. And at the same time, these parasites, which are stuck to the inner side of the blood vessels, start to induce a local inflammatory response, which induces the production of more pro-inflammatory cytokines and also chemokines, primarily CXCR3 chemokines. And these chemokines attract more lymphocytes, mainly CD4 and CD8 T cells, to the site of parasite sequestration, which only make things worse. Now, a few years back, when we established that CXCR3 was actually the main trafficking pathway mediating the recruitment of pathogenic T cells to the brain of infected animals, we asked the question, what happens if we actually block this chemotaxis pathway? Can we alleviate inflammation in the brain and prevent disease? And to address that question, we decided to focus on one of the 3 CXCR3 chemokines, which is CXCL10 or interferon gamma inducible protein 10, or IP10 for short. And the 2 approaches that we used was to develop monoclonal antibodies against IP10 and also to infect IP10 knockout mice. And basically what we found in those early studies was that both infection of IP10 knockout mice or treatment of C57 Black 6 mice with anti-IP10 neutralizing antibodies significantly protected mice from the development of cerebral malaria. And to confirm that actually the inflammation in the brain was reduced, what we did was a series of intravital microscopy experiments to visualize the microcirculation in the brain of these animals. And indeed, we could confirm that after treatment with anti-IP10 monoclonal antibodies was a substantial reduction in the number of cells rolling or adherent in the brain microvasculature compared to animals that had been treated with an isotype control antibody. So it was very good, and that was a really happy day in the lab, but this was the predictable part of the story. One of the things that we didn't see coming when we started a series of experiments was when we look at the parasitemia of these animals, and we found that both treatment with anti-IP10 or infection of IP10 knockout mice actually resulted in significantly lower parasitemia levels compared to the wild-type controls. Now, because this was an unexpected result, we started to look at this response in a little bit more more detail. And for that, instead of using wild-type parasites, what we did was to switch to some transgenic parasites that express luciferase. So when we inject these animals with luciferin, we can actually visualize that bioluminescence coming from the parasites and calculate what we call total parasite biomass. And why would we bother? Well, because as I said before, WEHI seems to sequester in different organs, so it has been argued that just looking at peripheral parasitemia might not be a very accurate estimation of the total parasite burden that the mouse is experiencing. So when we repeated those experiments with the luciferase bugs, basically we found interesting results. So both infection of IP10 knockout mice, as well as treatment with anti-IP10 monoclonal antibodies, resulted in massive 70% reduction in total parasite biomass, which started to indicate to us that IP10 is doing something bad. It's having a detrimental effect in the induction of immune mechanisms that are required to control the infection. Okay, so but the question is, where is all this IP10 coming from in the first place? And because both in humans and in mouse models of malaria, IP10 has been described to be a biomarker of cerebral malaria. Most of the attention has been put into brain tissue. So, for example, in humans, it's been shown to be found in cerebrospinal fluid of patients that succumb to falciparum malaria. And in mouse models, it's been shown to be produced by endothelial cells, astrocytes, and neurons. However, it is not clear whether the production of IP10 by this cell type is actually responsible for the processes that I described before. And interestingly, some work done here at WEHI by Danielle Stanisic in what is now the Division of Population Health revealed that IP10 produced by peripheral blood mononuclear cells is actually associated with severity to disease in an endemic area of Papua New Guinea. So this was the first indication that actually leukocytes that have IP10 could be responsible for the development of severe disease, which led us to come up with the first question, so what are the cellular sources of IP10 responsible for the development of severe malaria? And to address that question, we again used the Plasmodium berghei ANCA model, and we decided to do an irradiation chimera approach in which we took wild-type mice and IP10 knockout mice. Then these mice were irradiated and reconstituted with the opposite bone marrow, in that we end up with a mouse that has no hematopoietic, no IP10 secretion from hematopoietic derived cells, and another mouse that has no IP10 secretion from stromal cells. So these mice were then infected with Plasmodium berghei, and we examined susceptibility rates and parasite burden. And basically what we found that is, as expected, all the wild-type into wild-type chimeras died before day 9 post-infections, whereas the IP10 into IP10 chimeras, 80% of those animals survived the parasitic challenge. And it was actually these guys, so this is the black squares, the B6 into IP10. So this is the animals that secrete IP10 from their hematopoietic derived cells, the ones that develop severe malaria, and all these animals were gone within 2 weeks post-infection. And when we examined the parasitemia levels of these mice, basically what we found that once again, IP10 into IP10 chimeras had reduced levels of parasitemia compared to the wild-type into wild-type, and these IP10 into B6 animals were also quite efficient at controlling infection. And once again, these guys, the black squares, the ones that produce IP10 from the hematopoietic derived cells, the ones that had more difficulties controlling infection. So when they repeated these experiments using the luciferase bugs, and basically we found a similar trend in that once again, these animals, the mice that have IP10 secretion from their hematopoietic derived cells, could not control the infection as efficiently as IP10 into IP10 control chimeras. So we wanted to extend this approach and examine more specifically the sequestration rates of the parasite in the brain of these infected animals. And when we did that, basically we found a similar trend. As expected the B6 into B6 chimeras have high rates of sequestration of the parasite in the brain, significantly higher rates than the IP10 into IP10 chimeras. And once again, was the animals that express IP10 from their hematopoietic derived cells that could not control the infection and had higher sequestration of the parasite in the brain during infection. Now, as I said at the beginning of the talk, in this model, disease does not only result from the sequestration of parasites, but also from the recruitment of CXCR3 positive T cells, primarily CD8 T cells, to the brain of infected animals. We wanted to have a look at that, what happens with the different chimeras, and for that, what we did was to purify what we call the brain sequester leukocytes. These cells can be stained with fluorescent antibodies and analyzed by flow cytometry. And interestingly, what we found is that these mice, so these are the mice that are perfectly capable of secreting IP10 from stromal brain cells, like brain endothelial cells, or astrocytes or neurons. So these mice, despite having that compartment intact, basically failed to recruit significant numbers of CD8 T cells. And once again, where the cells, the animals that have IP10 secretion from their hematopoietic cell-derived compartment, the ones that recruited significant numbers of CD8 T cells to the brain of infected animals. Obviously, we wanted to determine, okay, what cells are responsible for all this, and to do that, what Eliza did in the lab was to take splenocytes and stain them with a whole range of different antibodies, so we could identify B cells, NKs, NKT cells, T cells, neutrophils, and inflammatory monocytes. And when did she do that? Well, she did this on day 3 post-infection, because our pilot data basically indicated that that's when IP10 levels peak. And then when Eliza determined the expression of IP10 in all these sorted populations by RT-PCR, basically she found that the vast majority of the IP10 was coming from inflammatory monocytes and neutrophils. Right, so this is the spleen, so what happens in the brain are actually these cells secreting IP10 in the brain. And to address that question, Eliza again purified the brain sequester leukocytes, and in this case she stained these cells with antibodies to be able to distinguish between lymphocytes, so as T cells and NK cells, and GR1 to identify monocytes and neutrophils, and also microglia. And in this case, what Eliza found is that again, T cells and NK cells failed to secrete IP10, and the vast majority of the IP10 was coming from the GR1 positive fraction that contains monocytes and neutrophils, with some contribution from microglial cells. Then we ask the question, is neutrophil and monocyte IP10 sufficient to induce disease? And to address that question, we pursued an adoptive transfer approach in which we isolated GR1 positive monocytes neutrophils from wild-type mice, and these cells were adoptively transferred into IP10 recipients, and then we infected the mice with Bergiae, parasitemia, and susceptibility rates. And basically what we found is that indeed adoptive transfer of wild-type monocytes and neutrophils into IP10 recipients significantly increased the susceptibility rates of otherwise resistant IP10 knockout mice, and this was accompanied by increased parasitemia levels compared to IP10 controls. Okay, so hopefully this very first part of the seminar gives you an idea that, surprisingly, is hematopoietic cell-derived IP10 the one that controls total parasite biomass, parasite and leukocyte sequestration rates in the brain, and susceptibility to cerebral malaria. And actually, it's monocytes and neutrophils, the main sources of this chemokine, that are responsible for this process. But one of the things that I didn't elaborate for now is what is the mechanism? How come that these mice are actually more resistant to infection? And one of the things that we found in our previous work is that this improved control of parasitemia that appears to take place in the absence of IP10 is associated with a preferential accumulation of CXCR3 positive T cells in the spleen of these infected animals that result from the blockade of trafficking of these cells to inflamed tissue. And we also know that this process requires CD4 T cells, because if we infect mice that lack CD4 T cells, like MHC class II knockout mice, with Bergiae, and we treat these animals with anti-IP10 monoclonal antibodies, we don't see any improvement in the control of parasitemia, as we usually see with wild-type mice or with mice that lack CD8 T cells. So we have done a fair bit of extra work trying to understand the mechanism on how these mice control parasitemia, but I'm going to go back to this during the second part of my talk, and you will see why it's going to make a lot more sense. So for now, I'm just going to leave you with a little model in which we have a human or a mouse for the purpose of this talk, because this has been in a mouse model. So this is the epithelial knockout mouse. Just remember this guy. I'm going to come back to this one a bit later. So when we put a malaria infection, a blood stage malaria infection, a number of things start to happen. On one hand, we have an immune response started to develop in the spleen, and on the other hand, the parasite is avoiding going into the spleen, starts to sequester in tissue. And when that happens, there is a first wave of innate cells like GR1 expressing monocytes that actually are recruited to the site of parasite sequestration. And these cells secrete a wave of CXCR3 chemokines, which attracts more pathogenic CD8 T cells to the site of parasite sequestration in the brain. Now, if we interfere with this pathway, a number of things start to happen. On one hand, we reduce the level of intravascular inflammation in the brain, and on the other hand, these CXCR3 positive T cells appear to accumulate at the site of origin in the spleen and mediate a range of different responses which are efficient to control the infection, which in turn results in lower sequestration of parasites in tissue and alleviates disease. But I guess that the concept that is trying to emerge from all this research is that having too much inflammation is actually a bad thing for the development of protective immune responses to infection. And that concept leads me to the second part of my talk, which is why is that it takes such a long time to become immune to malaria? Now, in the field, malaria is acquired slowly in a stepwise manner over a long period of time, in that immunity to severe malaria is usually acquired before the first 10 years of age, and after that, individuals might still experience symptoms of mild malaria, and it's only after around 20 years of constant exposure to the parasite that individuals living in malaria endemic areas develop what we call clinical immunity. So this immunity is not sterilizing, but prevents the development of clinical episodes, but substantially reducing parasitemia levels. We know that this naturally acquired immunity targets blood stage parasites, and we also know that requires antibody responses. However, despite the importance of antibody responses in the induction of clinical immunity, the cellular mechanisms underlying this very slow and imperfect acquisition of immunity have not been extensively investigated. And there's only quite a few, only few studies that actually looked at the development of memory B cells in human populations. Basically, what we have learned from those studies is that if we look at the development of memory B cells in areas of high transmissions, that is when people are exposed to the parasite all the time and experience very frequent clinical episodes, those individuals appear to have a delayed development of memory B cells specific for malarial antigens. On the other hand, if we examine these responses in areas of low transmission, where clinical episodes are rare, these individuals appear to be characterized as having a stable pool of memory B cells, even in the absence of frequent boosting. So this, as you remember from the first part of my talk, these clinical episodes are characterized by the production of pro-inflammatory cytokines. And this altogether, this information basically allow us to come up with a hypothesis that is that malaria clinical episodes, or more specifically, inflammatory response associated to these episodes, have a detrimental effect in the induction of B-cell-mediated immune responses to infection. And to address that question, we decided to stick to the Plasmodium Berger ANCA model because this is still the best available model that we have to look at severe malaria in mice. And to be able to appreciate the impact of inflammation, what we did was to examine immune responses in mice that were infected with Plasmodium Berger and in mice that were immunized with equivalent doses of non-replicating irradiated parasites. So in this system, we are able to analyze immune responses in the presence or the absence of severe malaria symptoms and the inflammation that is associated to these symptoms. Now, because Bergia induces a lethal infection in black 6 mice, what we have to do in this system is to treat mice with anti-malarial drugs around day 5 post-infection and the onset of disease symptoms to be able to measure antibody responses or cellular responses at much later time points post-exposure. And in the very first series of experiments, what we did was just to look at antibody responses. And indeed, what we found is that in the absence of inflammation, mice that had been immunized with irradiated parasites mounted a parasite-specific antibody response 5 to 6 times higher to the response obtained from mice that experienced an active infection. So we said, okay, this could be an interesting system to examine these responses. Let's have a look at the cells. And before I do that, I'm going to take just 1 minute to remind you all how B-cell responses are generated in the lymphoid organs. So very, very briefly, this process starts when an activated B-cell, when a naive T-cell actually becomes activated after encounter with antigens. So this activated B-cell has 2 different options. It can either become a short-lived plasma cell, and these cells actually secrete the very first wave of antibody that is required to control the infection, or it can migrate deeper into a follicle and establish what we call a germinal center. So these B cells in germinal centers, together with the help of T follicular helper cells, undergo the process of affinity maturation, by which they undergo hypermutation in their immunoglobulin genes. And then the cells that leave the germinal center secrete immunoglobulin of much higher affinity for antigen. And the 2 populations of experienced B cells that leave the germinal center are actually these long-lived plasma cells that usually go to the bone marrow, or memory B cells that stay in circulation and become rapidly activated after second encounter with the antigen. So we have actually analyzed all these different populations during severe malaria infections, but in the interest of time, I'm going to focus today only in the germinal center responses. Now, to basically look at the impact of severe malaria infections in germinal center reactions, what we did was to, again, infect a group of mice with Plasmodium berghei and can have another group immunized with equivalent doses of irradiated parasites. And then what we did was to prepare spleen sections at different time post-exposure, and these sections were stained with anti-IgD antibodies to look at the B-cell follicles shown in green, anti-C3 shown in blue to identify T zones, and anti-GL7 in red to identify the establishment of germinal centers. And as you can see, on day 6 and day 9 post-immunizations, these animals that receive irradiated parasites start to respond and establish really robust germinal center reactions in the spleen of these animals. And in contrast, animals that are experiencing an active malaria infection were completely unable to establish germinal center reactions in the spleen on day 6 and day 9. And it's only from day 12 onwards, so this is when the animals have completed the anti-malarial drug treatment and completely cleared infection. These mice start to develop germinal centers in the spleen, and this response keeps improving on day 24 post-exposure. So we then wanted to look at the different populations in germinal centers in more detail, so for that we use flow cytometry. And in this case, we look at the frequencies and absolute numbers of germinal center B cells defined as high expression of GL7 and low expression of CD38. And basically what we found is that both the frequency as well as the absolute number of germinal center B cells was significantly reduced in mice that were experiencing an active malaria infection compared to controls immunized with irradiated parasites. We then look at follicular helper T cells, and basically we find a very similar result. Mice experiencing a Berghia ANCA infection had significantly lower percentages and numbers of follicular helper T cells defined as CD4 T cells expressing high levels of PD-1 and CXCR5. And Victoria, that was a PhD student in the lab working in this project, one day comes to the office and tells me, Diana, I think that there is a second population of follicular helper T cells in the infected mice. And she was absolutely right. What Victoria found is that during infection, there was a second population of follicular helper T cells that express actually intermediate levels of PD-1 and CXCR5. And these cells had actually been previously described in the literature and appeared to be precursors of the mature PD-1 high, CXCR5 high follicular helper T cells. And in further analysis, where Victoria enumerated these cells in the spleen, basically what she found is that despite this clear defect in the absolute number of mature follicular helper cells found in response to infection, the total number of pre-follicular helper cells, so this is the intermediate population, was virtually identical between infected and immunized animals, which led us to postulate that basically infection was impairing or inhibiting the proper follicular helper cell differentiation. So before getting too carried away, we wanted to actually confirm that these cells were follicular helper cells. And for that, we look at the expression of BCL6, which is the master transcription factor required for the differentiation of follicular helper cells. And indeed, what we found is that when we examine all the BCL6 positive, CD4 positive cells, these cells only express intermediate levels of PD-1 and CXCR5 after infection with Plasmodium berger ANCA, and the two different subsets, the precursors of the mature cells, could be found after immunization with irradiated parasites. So we took this one step further, and we wanted to see if these cells were actually capable of producing IL-21. And for that, we used an IL-21 GFP reporter mouse that was generated here at WEHI. And indeed, when we get it on the CD4 positive T cells expressing IL-21 in 6 days after an active infection with Plasmodium berger ANCA, we found the vast majority of these cells express only intermediate levels of PD-1 and CXCR5. Whereas when we did this same analysis with cells from immunized animals, we found that both mature and immature cells were present more or less at a 50-50 ratio. Now, because there was a couple of papers starting to come up in the literature indicating that the cytokine environment could actually modulate the balance between Th1 and TFH cells, we wanted to examine if these follicular helper cells generated in response to a malaria infection share some of these Th1-like features. And indeed, what we found is that the expression of the transcription factor TBET was significantly higher in follicular helper T cells coming from infected mice compared to those ones coming from mice immunized with irradiated parasites. And moreover, when we examined the CXCR3 expression, we found that only 60% of the follicular helper T cells for immunized mice express CXCR3. Almost every follicular helper T cell coming from an active infection express these inflammatory chemokine receptors. So what we have so far is that infection is primarily inducing a population of follicular helper cell precursors that share some features with Th1 cells. Now, you might remember from the first part of my talk that interferon gamma and TNF were two crucial cytokines involved in the development of severe malaria syndromes. So we wanted to examine whether these cytokines were also playing a role in the inhibition of germinal center responses. And to do that, what we did was to infect black 6 mice, and these mice were treated with neutralizing antibodies against TNF and interferon gamma or isotype controls, and we looked at a range of different responses. And basically what we found is that a neutralization of these two pro-inflammatory cytokines significantly increased the number of follicular helper cell precursors, primarily on day 6, but not so much on day 9 post-infection. More importantly, the absolute number of mature follicular helper T cells was significantly improved upon neutralization of these two pro-inflammatory cytokines. And when we look at germinal center responses in the spleen, indeed we found that when we neutralize interferon gamma and TNF, this improves overall splenic architecture and also allows establishment of germinal center reactions as early as day 6 post-infection. And this is also accompanied by an increased number of germinal center B cells in the spleen and improved parasite-specific antibody responses to infection. So we next wanted to determine whether actually the presence of the transcription factor TBET was affecting the capacity of these follicular helper cells to fully differentiate. And for that, we pursued a mixed chimera approach in which we took mice, and these mice were irradiated and reconstituted with a 1-to-1 mixture of congenitally marked wild type and TBET knockout bone marrows. So then these chimeras were infected with Plasmodium bergheri enca, and we actually could have a look at the differentiation of follicular helper T cells in the 2 different compartments. And basically what we found is that in the TBET knockout compartment during the infection, these cells gave rise to significantly higher frequencies of mature follicular helper cells compared to wild types, and this is defined either by high expression of BCL6 and CXCR5 or PD1 and CXCR5. And when we look at the consequences of this, and we examine germinal center responses in these mixed chimeras compared to all wild-type control chimeras, we found that these animals that have 50% of TBET knockout bone marrow have increased number of germinal center B cells and allowed a really efficient establishment of germinal center B cells in the spleens compared to controls. And moreover, we saw that this was also accompanied by increased significantly higher numbers of titers of parasite-specific IgG1 but not IgG2A antibody responses to infection. Now, remember this guy. So this was the IP10 knockout mice, and I said that I was going to come back to this guy, the second part of my talk. So this mouse is really happy because it's highly resistant to malaria, and all what we knew before is that this increased resistance was associated with an increased accumulation of CXCR3 positive T cells in the spleen that resulted from the inhibition of trafficking to inflamed tissue. Now we know that all follicular helper T cells during malaria actually express CXCR3, so the next obvious question was, this mouse has more follicular helper T cells? And yeah, it does. So the absolute number of both pre-follicular helper cells and mature follicular helper cells is increased in IP10 knockout mice compared to wild-type controls. But despite this increased number of follicular helper T cells, we could not detect an improvement in germinal center reactions in these animals, at least on day 6 post-infection. Probably we need to examine what happens at later time points post-infection. But follicular helper T cells are not only important for the establishment of germinal centers, also important to provide help for extrafollicular responses. In particular, this population, the precursors that express intermediate levels of PD1 and CXCR5 have been shown to be critical for the induction of extrafollicular responses. So then we look at what happened with the plasmablasts in these mice, and indeed what we found is that both the frequency and absolute number of isotyped switch plasmablast was significantly higher in IP10 knockout mice compared to wild-type controls, and this was also accompanied by increased antibody titers to infection compared to wild-type animals. So hopefully I managed to give you a feel that severe malaria infections inhibit germinal center reactions, and anti-follicular helper cell differentiation appeared to be compromised by these acute malaria infections. And moreover, the expression of the transcription factor TBET impairs T follicular helper cell differentiation in response to infection. And more importantly, the concept that emerges from all this is that the same inflammatory factors that are responsible for the development of severe malaria syndromes appear to play a detrimental role for the induction of B-cell-mediated immune responses. So just to wrap up the second part of the talk, I'm going to leave you with this little model on how germinal center reactions occur or don't occur during malaria infection. So unlike a normal pathway of follicular helper cell differentiation in which a naive cell becomes activated and when it's surrounded by the right signals upregulates BCL6 and CXCR5, and this allows the migration into the germinal centers, what happens in malaria is that due to the strong pro-inflammatory environment that we have in the spleen of these animals, we end up with a weird follicular helper T cell that co-expresses BCL6 and TBET, and in addition of expressing low levels of CXCR5, expresses high levels of the pro-inflammatory chemokine receptor CXCR3. So this follicular helper T cell is not very helpful for inducing germinal center reactions, and moreover, because it expresses high levels of an inflammatory chemokine receptor, it might be attracted to migrate away from the lymphoid organ and probably misses an opportunity to provide help for antibody responses completely. All right, so the last 10 minutes that I got left, I would like to switch gears a little bit and hopefully give you a little flavor of some more translational work that we are doing in the division, which aims to identify antigenic targets of naturally acquired immunity to malaria in order to inform in the design of novel anti-malarial vaccines. And as I said before, antibodies are important for protection from clinical malaria, but the targets of that response are not completely characterized. And that's one area that we are really interested in the division altogether. So this cartoon here represents the structure of the merozoite, which is the extracellular form of the parasite before it's going to invade the red blood cell. So you can see there are a lot of proteins in the surface that could be potential vaccine candidates. So we have these two families, EVAs and RHs. So these proteins are involved in the invasion of the parasite into the red blood cell, and they're actually expressed onto the surface of the parasite just before the invasion event. Then we have some other proteins which are constitutively expressed on the parasite, which we call merozoite surface proteins, or MSPs. Many of these proteins have a GPI anchor, and some other proteins don't have a GPI anchor and appear to be associated to one of these GPI anchor proteins to be exposed on the parasite surface. And today I'm going to be talking about two of these extrinsically associated proteins that were recently characterized in the Hansen Lab, called MSP-DBL1 and MSP-DBL2. And basically, the approach that we have in the division to determine whether these proteins are targets of naturally acquired immunity and therefore good vaccine candidates is to examine the immune responses to these proteins in malaria endemic areas. And today I'm going to be focusing on one cohort study that was conducted in Papua New Guinea by Ivo Mueller many years ago now. So this is the MUGAL cohort study, which enrolled around 200 children between 5 to 12 years of age. So all these kids were treated with anti-malarial drugs at enrollment, and they donated a sample of blood. And then the kids are followed up for a period of 6 months for reinfections or clinical episodes. And then what we do basically is to try to associate immune responses at enrollment with disease outcomes. Now, a little bit about MSP-DBL1 and 2. Why am I interested in these proteins? Well, I think they're really interesting because they share features from two completely different families, protein families in B. falciparum. For example, they have this SPAM domain, which is a very conserved domain, usually found on the MSP3-like family. And within the same molecule, they express this DBL domain, which is highly polymorphic and is usually found in proteins that are involved in the invasion of the into the red blood cell, like EVA140 and EVA175. What we know about these proteins is that they actually bind to red blood cells via their DBL domains. And we also know that there is this leucine-like zipper adjacent to the SPAM domain, and that this leucine zipper appears to be required for oligomerization of these proteins. And some recent biochemical work in the Hansen Lab actually described that MSP-DBL1 and 2 form a big complex with MSP1, which is one of the GPI anchor proteins, and MSP6 on the merozoite surface, in that MSP1 appears to be like a platform to display the MSP DBLs for binding to a yet-to-be characterized receptor on the surface of the red blood cell. Now, when we look at antibody responses against MSP DBL1 and 2 in this cohort, basically what we find is something like this. Responses are really, really heterogeneous in that we have high responders, medium responders, and low responders. And that allows us to break up this cohort in terciles and basically analyze the time that took to each different group to become reinfected again or to develop a clinical episode. And when we do this analysis, basically what we found is that individuals that had high or intermediate levels of anti-MSP-DBL1 or anti-MSP-DBL2 antibodies took a lot longer to become reinfected or to develop a clinical episode in this case, compared to the individuals who have low levels of anti-MSP-DBL1 and 2 antibodies. So we wanted to take this a little further and try to determine which one of the different domains within the MSP-DBLs were responsible for this effect. So for that, we did antibody responses specific for DBL spam domains or the spam domain containing the leucine-like CEPA. And this analysis is showing hazard ratios. Hazard ratios are basically giving us a feel of the probability of something happening. When a hazard ratio is lower than 1, that means that it's a reduced risk of that response, and when the hazard ratio is higher than 1 means that it's an increased risk. And as you can see, what we found here is that having antibodies against all these different domains of MSP-DBL1 significantly reduced the risk of acquiring a clinical episode. When we look at these responses with MSP-DBL2, basically what we found is that antibodies directed only against the conserved spam domain were actually associated with protection from clinical malaria. And finally, what we did was to try to determine the effector mechanism by which these antibodies protect against a clinical malaria. And to do that, what we did was to purify antigen-specific from the responder individuals in the cohort study. So this human antibody then was used to incubate with ethidium bromide labeled parasites, and these opsonized parasites were then incubated with a monocytic cell line, and that allows us to basically measure the phagocytosis mediated by antibodies. And basically what we found is that both anti-MSP-DBL1 and anti-MSP-DBL2 have strong opsonizing capacity, which actually titrates in a dose-response manner, reaches a peak at really low concentrations like 5 micrograms per ml. And then in a final experiment, we wanted to determine whether these human antibodies could also inhibit the ability of P. falciparum to grow in vitro, and the answer is yes. So when we set up these growth inhibition assays, basically we found that both human anti-MSP-DBL1 and 2 significantly inhibit P. falciparum growth in vitro compared to IgG from non-exposed individuals. So hopefully that gave you a little flavor of what we can do in the clinical samples. And in this particular case, we found that antibodies directed to MSP-DBL1 and 2 are associated with protection from clinical malaria, and that both polymorphic as well as conserved domains of this molecule are the target of this protection. And MSP-DBL1 and 2 antibodies appear to have strong opsonizing capacity and also inhibit Plasmodium falciparum growth in vitro. And more important, MSP-DBL1 and 2 are targets of naturally acquired immunity to malaria, and as such, they could be considered as an important, new important vaccine candidates. Okay, so that's all what I have to say. But before I go into the acknowledgement slides, I'd like to take one more minute just to give you a feel of the future directions that we have in the lab for the next few years. So we are hoping to continue our rodent malaria program in collaboration with immunology and molecular immunology divisions. And we would also like to extend our translational malaria program through new collaborations with Ackman Institute of Molecular Biology in Jakarta, in Indonesia. And the idea of these translational studies is to keep working on the identification of targets of naturally acquired immunity, to keep informing the design of an anti-malarial vaccine, and also to start translating some of our more recent findings in the mouse model of malaria. For example, to look at whether a defect in the T follicular helper cell compartment is also responsible for the delayed acquisition of immunity to P. falciparum in the field. And the Eichmann Institute has a capacity to conduct field work in different islands within Indonesia, and the work that we have started already is in this in Papua, in this region called Timika, which is in the south coast of Papua. Over the past 2 years, I had opportunities to visit Indonesia a few times and had wonderful meetings and conversations with scientists, not only working in malaria, also working in a different range of infectious diseases like hepatitis, dengue, or TB. And all these scientists are really interested in the type of research that we are doing here at WEHI. So if you are thinking about translational research in these areas, please come to see me. All right, so that's all what I have to say so far, and I would like to start acknowledging a long list of people, starting with current and past members of my lab. Lisa Ioannidis, a postdoc in lab, did all the IP10 project, and Victoria started the T follicular helper cell project in the context of her PhD, which is now being continued by Anne Lai. And Chris Chu did the analysis of antibody responses to MSP-DBL1 and 2 that I share with you today. Apart from my lab, I would like to thank the Kauman lab, in particularly Alan Kauman, for giving me the opportunity to set up this malaria immunology program in the division. His support over the past 4 years has been incredible, and I'm truly thankful for this opportunity. I also like to thank members of the lab like Julie, Jenny, Tony, Clara, Lynn, Wilson. They all gave me so many reagents, proteins, and antibodies for work that I presented today and other studies I didn't have an opportunity to share with you today. The Pellegrini lab, especially Mark, for being a great mentor, and Simon also provided some mutant mice for the analysis of B-cell responses. Ivo Müller from the Population Health Division and Peter Siever for access to aliquots of the Mughal cohort study. Danica Hill did the opsonization studies that I showed you today, and many thanks to Connie Lewy-Swen for statistical analysis. All the analysis of B-cell responses in the rodent malaria system was done in collaboration with immunology and molecular immunology divisions. I'd like to thank Lynn and David for mice and protocols and discussions, things that I didn't even have a chance to present today. Special thanks to Stephen Nutt, same thing, a lot of advice and reagents. And my very special thanks go to Alex Callis, that was my partner in crime in the follicular helper cell story that I presented today. Thanks to the Imaging Center, Kelly and her team, the Fax Lab and the mouse rooms, particularly Liana Makiewicz, that takes care of our mice. Catherine Nee was initially a PhD student in the lab, and she actually initiated most of the projects that I described today. She's now at the Burnett. And thanks to Rintis and her team in the Eichmann Institute in Jakarta. And all my research is funded by the NHMRC. Thank you for your attention.
Thanks, Diana. Questions?
Trying to look for antigens that produce natural immunity. We look at an antibody titer versus clinical outcome. How do you dissociate that from just a generally better immune response? Don't you have to normalize it to more general antibodies?
Yeah, I mean, I didn't have much time to elaborate on that, but all those results are adjusted for a list of different confounders, which include the geographic location and age. So those hazard ratios that I was showing are actually adjusted by all those confounders that we usually find in the cohort study.
Yes, but that's not what I asked. So if a person has produced a number of antibodies against malaria and they are therefore protected for longer, then the antibodies to your specific antigens, the MSP, DBL proteins might just be one of many, and so the hazard ratio has to be adjusted for. You have to isolate it from a more general immune response, I would have thought.
Yeah, I mean, there are different responses. Some, when you do this analysis with a range of different responses, some reveal an association for protection and some not. You can do like a also multivariate analysis that basically gives you an idea of integrating different responses and how that enhances the protection. For example, that good example for that is if you're looking at the capacity of antibodies directed against parasite invasion ligands. Parasite is very smart and utilizes different doors to get into the red blood cells. So our working hypothesis is that just having one antigen using a vaccine as an invasion ligand won't be efficient because the parasite would utilize other doors. So in other words, what I'm trying to say is that hopefully you need to get an immune response that targets several different antigens to be able to have more comprehensive protection.
So it's very nice, but there are a number of responses that generate lots of gamma interferon, to which there's normal memory produced, and presumably they also induce a lot of T-VET in their T-follicular helper cells and upregulate CXCR3. So it doesn't have the same outcome. So do you think there's something in addition happening in the malaria infection to just gamma interferon and T-VET upregulation in the T-follicular helper cells?
We don't know. That's all we have so far. We know that they express that, and we know that these cells that express low levels of CXCR5 are not very good at getting into germinal center reactions. So they might be, if they stick around, be efficient to provide help for extrafollicular responses. But because they are also expressing CXCR3, chances are, as I was saying in the discussion, that if they are exposed to very strong chemotactic stimuli, they might migrate out of the lymphoid organs and miss out completely on the opportunity of provide help for antibody responses. We don't know if there is anything else apart from that the upregulation of T-VET. We haven't examined what other things they do. We know that they produce higher levels of interferon gamma and TNF than the cells from immunized animals, but they seem to produce normal levels of IL-21. So in theory, they should be good helpers, it's that they are not localized in the right location.
Okay. Yeah, Andreas.
So the difference in the susceptibility between black 6 and bulb C mice, is that explained by differences in IP10 production between the two strains?
The difference is explained not only by IP10, by also by the amount of gamma that is produced in these mice, and that IP10 is actually induced by interferon gamma. So some work that we've done in the past basically shows that the expression of NK and NKC receptors, which modulates the output of gamma, actually contributes to the differential susceptibility of bulb C and black 6 in this model. So IP10 is probably a bit downstream of that gamma. Yeah.
Okay. Dave, that was the last one.
With the IP10 knockouts made on 129 ES cells and then back-crossed, or was it?
They were back-crossed 9 times, I think. Yeah. Joanna Groom is in the audience. She should know. No, she's not.
Yeah.
Oh, 9. Is that accurate?
Yeah, they're very well back-crossed.
Yeah.
But they do express CXCL11 in addition to the knockout because of the 129.
Yeah.
Okay, let's thank Diana.